All right, we've reached the third lecture on regulation of gene expression. I'm going to cover the next few steps here. I'm not going to cover them in the detail that we did for the control of transcription, but we certainly could. There's a huge amount of information that's coming forth about each one of these steps. But again, we just kind of want to work us through the process and talk a little more general about it. So up to this point, we've talked about chromatin remodeling as one of the methods that we can control and regulate gene expression. We spent quite a bit of time talking about transcription and all the various levels at which that can occur, and these idea that you've got enhancers and silencers and all these repression ideas that can come along. What we want to do in this lecture is cover the next three. We want to talk about RNA processing, we want to talk about translational control, as well as post-translational control. And remember that, as we work down this model, the energy required for the cell to do this, it becomes greater and greater and greater. If we block it at the chromatin level, we spent almost no energy except for the energy to produce the blocking agents or control it. If we move to transcription and we control it there, we're using more energy because of all that transcriptional complex that's needed. If we move to the next level, more and more energy had to be invested by the cell. So what we find is while each one of these steps occurs, okay, they're only those proteins that we really need to have quickly are maintained at one of these translational levels or post-translational levels. If we don't need it in a hurry, then we can block it way back up here, okay? But even if we don't unblock it, every stage occurs through here is actually a regulatory stage. So you will, for the same protein that you're going to produce, you will have control occurring at the chromatin. You'll have some obvious control that has to occur at the transcriptional level. You'll have control that occurs at the maturing level, and you'll have translational level control. Every one of those has to occur for every protein. Okay, or does occur for every protein. So again, even though it's energetically expensive, and you kind of think of it in terms of where would we want to put our supply, even if you do start from ground zero, every stage involves energy and regulation in itself. Okay, so let's take a little bit of look at RNA processing. Obviously, we know that when we post transcriptionally, we modify the message, at least the messages that we're thinking about here. And we can do that in various ways. We can control the degradation of the system by adding these poly A tails, right? Increasing the length of the tails themselves will control how quickly the message can be, how quickly it will disappear, or how long, and conversely, how long it might maintain itself out in the cytoplasm. If you remove the cap, then you can definitely control that end of it, and whether you're even going to have the official formation of the translational structures that are needed, the ribosomes, etc. But another thing you can do, obviously, is you can control splicing. And it turns out we've talked about alternate splicing and all the things that have to occur there, but the spliceosomes and things, alternate splicing is definitely an area where you have control. A good example, and the one that the book uses, is talks about how we actually get male and female fruit flies. For our purposes, we've always said if you XX, just like a human, you get a female, XY, you get a male. So we just said, basically, it's the ligase mode of sex determination, XXXY. Well, how does that really occur? Well, it actually turns out it's a bit more complicated, because what happens is this. When you have two X chromosomes, there is a STI gene that's associated with that. And in the embryos for that, this gene is activated, okay? The gene produces a protein that is involved in helping to splice other proteins, okay? And eventually it involves the splicing of this DSX protein itself, this RNA, rather, not this protein, but the splicing of that produces finally this protein. If that protein is produced in a functional form, okay, then the types of protein that is produced, then you end up with a female fly. And if this gene is downregulated, right, which it is in the case of XY individuals, you don't get the same splicing. You get a dysfunctional intermediate protein, okay, and you get a differently spliced or differently formed up final protein. So you end up being males. And kind of take a look at how that works. Here's the intermediate protein here, the triprotein. This is the pre-mRNA that's involved, okay? Obviously, there are introns and exons, and there are multiple splice sites. Okay, if the striding protein has been produced, then it is involved in the lariat formation and the splicing of it. Okay, now what it does is it cuts out this B area here, this area, and by removing that area, internally in that area that's now an intron, which would be an exon otherwise, right, is an early stop code. There's a codon in here that would stop it. Well, if you remove that and you work through the system, then you now have the splicing of A, B, and D units together to make a functional protein, a trial protein. And it's functional. Once it's functional, you end up making females. It diverts you along that female line, much in the same way something like estrogen or something might push us toward being female in humans. Okay. If you don't have this protein available, then you splice it. You actually remain, the B part remains in the system. You don't actually get C, but you get D. So you keep B as an alternate splice form. The problem with that is that there's a stop codon in the middle of B, and so you get a stop during translation. Stops early, and the stop protein is non-functional because it's too short. Right? Essentially, it doesn't have all the components. If it's too short, in this case, or it doesn't have the correct number of amino acids, it can't fold correctly, it can't go into its tertiary configuration, and so it's a non-functional protein. But what that ends up doing is producing males. Okay, so in this way, we see that we can, how you splice it, and what regulates that splicing are other proteins that become involved, particularly in this case in the spliceosome region, that control this actual message itself. Right? Now, another thing you can do is we've learned recently that there's things known as RNA interference. And there's a number of ways that this can occur, but you can make a message, you can mature it all you want, get it out usually into the cytoplasm. And then there are things that will interfere with whether that message is actually going to make it to being translated or not. Okay. Now, a good example of that are what are known as small interfering RNAs. Okay, these are RNAs that are made as double-stranded RNA. Okay, they're red, so they're red. We carry them in our genome. Okay, there's a special enzyme known as dicer which recognizes these and cuts them up into small pieces. Okay. Other proteins come into play, a complex of proteins, and they associate those sliced up pieces, now single-stranded RNA, with message. Okay? When they are associated with that message, okay, when this RNA is associated with that message, then you get cleavage. That cleavage usually occurs somewhere upstream from the poly A tail. And now you've got an open-ended RNA message that's available to be eaten up, essentially what happens. Now, these are important in normal process because we need to get rid of these messages, right? So this is one of the ways that messages are degraded is by these splicer proteins that come along, these RNAs that are made available. And so once a message is used and you want to degrade it, you'll have a hybridization of one of these small pieces of RNA to your message. You get cleavage, and you'll get that'll signal degradation of the message itself. Okay, or if you get too many messages, you can have it just producing messages, and the cell says, no, I don't really need those messages. It'll start doing this to them as well. So this is called interference. It can be a positive thing in that this is a way we get rid of many of our messages in a normal process. It can be a negative thing if it overruns the system and you're trying to make the message, but it never gets to the translational process because it got cleaved out. Then you end up with a problem. So it can lead to normal processes. It can lead to other types of processes. But it certainly is a regulatory area. Okay, think about it. We don't want this message sitting there forever, or maybe we do, okay, but if we don't, then we've got to get rid of it some way or the other, and one of the ways we can do that is either by the poly A tail being eaten away or by sending in these other signals that control it. So this is one of the ways that we see that happening, okay? And then obviously at the translational process, lots of things have to happen. I said there was this whole cascade of things that had to occur for transcription to happen, right? You have signaling and formation of the groups and the transcriptional complex and attracting in RNA pol. Well, translation is the same thing, okay? In translation, we have to be able to attract a small subunit of the ribosome. We have to build the large subunit. We have to pull in the tRNAs that we need. All of that has to happen. Well, that all happens by the fact that you often have other proteins that interact with the 5' end. I said that cap was important for sort of recognizing it, but there are all these interacting proteins that must play a role. There's shapes and forms. This is just one example here. They don't all form hairpins, but there can be formations. All sorts of things can happen upstream from the actual code that we're going to use, where the ATG start code is, right? It has to be, we have lots of things that go on up there, okay? And all of this area controls whether translation can even occur or not. If you don't have these proper interacting proteins, or you don't have any of these things firing in and the right kind of restructuring, you won't get translation. It can be controlled. Other things that have to happen is known as subcellular localization. You don't want to make this protein over here if you need it somewhere else, right? They try to make them close by to where you need them. They're not all just made in a factory or near the endoplasmic reticulum. They're moved around. So if I need one that's going to be associated with a mitochondria, then I will move it to the areas that might have mitochondria. If it's going to be a nuclear membrane-bound protein, then I'll subcellularly localize it to and produce it near the nuclear region or move it. And a lot of that occurs over here on the 3' end down here before you get to the poly A tail. There's some coding that goes on down here to tell you where you want to go. There's also stability coding and all those other things that can go on. So the point of this is, just like transcription, translation itself can be highly regulated as both where it happens, how quickly it happens, whether it happens at all. When the message can just sit there, just like in when we produce eggs, oogenesis, we often put down message that we're going to use for the first events once the fertilization occurs, and those messages sit there rested in translation until the fertilization event occurs, and this cascade of events come along and say, yep, start using that message. Let's go. We need the proteins that are going to be produced from that. Okay, so that's one of the areas, and a very complex system. Subcellular localization is a whole semester of ideas in itself. Certainly translational control, transcriptional control, translational control. In this case, either one of those are semester-long structures and discussions that go on. Okay. All right, and then finally, obviously, we need post-translational control, because remember, once we translate, all we produce is a polypeptide. We've still got to make that into a protein. We've got to do various things to it. We've got to form secondary structures, level structures, tertiary level structures, and maybe even quaternary level structures. And in doing all that, lots of things can happen, okay? One example that we see is that you can have these disulfide bonds occur, so that you get folding of the system, and there can be all sorts of cleavage. We can take. I mentioned a number of times that the polypeptide itself, even though MET may be the first amino acid put down because of the start codon, it's often cleaved off. Okay, it's often removed, and it's a signal, and other things are added on to signal, okay, this is a protein that needs to go into the nucleus, or no, it's not a protein that's going to go there, or it's got to be membrane bound or something else. Okay, a good example of this probably is insulin, small polypeptide produced. Okay, once it's produced as a form, you end up with these areas called chains. That's because it simply starts to fold. What you end up getting are these disulfide bridges that form. And you actually do, again, cleave off the MET end, okay, get rid of that. Once these disulfide bridges form, you get proteolysis, which means protein is cut, right? So essentially splicing of the protein. In this particular case, you cut out a large section of the protein itself, okay? And then the mature insulin, the insulin that's actually going to be the hormone, it's going to have the effect, looks something like this. Okay, it's not a continuous piece of, or it's some other structure that's bonded together by these sulfide bonds, and it's been highly modified because you've cleaved off a number of these proteins, these parts of the peptide, the amino acids. When cleavage doesn't happen right, you don't get the right insulin, and if you have insulin in various different uses, you may in fact have different, but this is what makes it into a small functional protein that can serve as a hormone, right? And then finally, there's lots of other things that can happen to it. One of the things we can do is we can attach lipids We can attach areas that make it lipid-philic, so that it's going to be there. That's particularly important for things where there's going to be a membrane-bound protein or membrane-associated protein. And there are at least 5 different kinds of lipids that can be added that are commonly added to proteins to signal which membrane and which areas they might actually attach to, or whether they're going to, sometimes adding lipids instead of going to membranes, it makes that protein functional to interact with another protein because of a lipid interaction that can occur because of the types of bonding that can happen there. So there's a number of different types of lipids, and depending on what that protein, where it's going and what it's actually going to do, you can have this formation. So if you don't want that protein, if it's a receptor, and you don't want it to be moved up to the area where this might be on the external surface of the cell, the membrane. You simply don't have a lipid component added to it. It just sits there. When you need it, a lipid component can be there. It can move up. Okay. There are other things that can happen. There can be phosphorylation, very common. There can be ubiquination. Ubiquin can be, that's usually a signaling that you want to move it out and get rid of it, but it can be a turn on, turn off system. But we can also phosphorylate it, causing that final attachment of phosphate groups and other things that can, particularly is very, very important in enzymatic proteins, so making them active. So we can have them there, and we may not be an active enzyme until we phosphorylate it, then it becomes active, or sometimes we phosphorylate it, it becomes non-active. It just depends on the form. So there have to be enzymes that come in and phosphorylate, phosphorylating enzymes. So they're proteins that are produced over here as well. So my point of all this is that two basic things to point out. One, regulation can and does occur at every level, okay? And in that, obviously, there's an energy cost as you move through the process, okay? So the first thing is it can and is controlled at multiple levels. It can be very fine-tuned as how the process works. Secondly, it's a very complex set of systems, and we're only learning about some of the ways that it works, the regulation and control. It's certainly been the last 15 or so years that we really started to understand gene regulation. I know when I first started teaching genetics, people would say, can we clone a human? And I say, I don't know, we probably can't clone a human, blah, 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 because, well, sure enough we can. And I said, well, but, we can't really do it right because we don't understand regulation. Well, we're starting to understand more and more about regulation and how to turn it and how to do it correctly. And this is one of the really key areas of the system. Okay. All right, so now we've covered regulation. I think enough for us. Certainly matched up to things in the textbook and others. Gives you a real handle now on one of the more important areas of how this system works. Again, once you do cell biology and you talk, and maybe even molecular genetics, you'll get much more into the regulatory control, cell signaling, how the response occurs, and that. But now you at least have a basis for some terms you'll see and understanding the background and basic ideas of what might go on to get a gene turned on and to get a protein produced.